I don't know how much enjoyment you had last night and how awake you are this morning, but there's quite a lot to unpack here. But I've been asked to talk about something called Manifest, and I'll try and take it slowly and obviously answer any questions. So Manifest stands for Multi-Omic Analysis of Immunotherapy Features Evidencing Success and Toxicity. Wow, okay, so let's stop there. Let's try and unpack that and help to understand what that's all about. And first I should say that I'm a collaborator in this project. Samra Turajlik, whose picture I put on that slide, is actually the person who's pulled this together. And unfortunately she can't be here today, so I'm speaking on her behalf. So immunotherapy, what's that all about? Now, in our body, we have T cells that are really important in fighting abnormal things. So here you can see the T cells understand how to distinguish between self and non-self. And in the diagram here, you can see a picture of what I would say self, our normal organs. The T cells leave our normal organs alone. But if there's a pathogen, coronavirus, those kind of things that enter our body, the T cell gets to work. It recognizes the non-self. Now, can it do that against tumour cells? The problem about tumour cells is although they're abnormal cells, they are derived from normal cells, and that makes it difficult for the T cells to recognise them. And also the tumours have ways of suppressing our immune system. So all this immunotherapy that you hear about is all about trying to harness that immune response. And in the last decade, things have changed enormously, as some of you, I'm sure, will know. And the cartoon on the right-hand corner there is a cartoon of a T cell. And a T cell basically is covered with lots of receptors that engage with the surrounding molecules. And these guys, whose pictures are there, Jim Allison and Tetsuko Honjo, won the Nobel Prize. That's an amazing thing, because of the work that they did to discover the importance of particular receptors, the CTLA4 and PD-1 receptor. And if you could block those receptors are keeping those T cells in check, stopping it going out and killing, hopefully, our cancer cells. If you can block them, you can take the brakes off the immune system, as we describe, and get those T cells to get about their business and try and identify cancer cells and destroy them. So that's the concept of the immune checkpoint inhibitors that are now very much part and parcel of how we manage many cancers. Now, this is a survival curve. When we do clinical trials, we monitor people, and they are essentially numbers in large-scale trials. There's nearly 1,000 patients that took part in this trial that evaluated the checkpoint inhibitors that we now recognize as standard of care, ipilimumab, nivolumab plus nivolumab. And at the start of the trial, when we start patients, everybody's alive, so you can see that everybody is at that number 100%. But over time, unfortunately, people will die of their disease. But what you can see here is that these curves plateau off. And that suggests that there's a proportion of patients who are actually surviving despite their cancer because they're responding to their treatment. So that's really important. I've got a curve here as well, haven't I? So you can see that up to 50% of people now are surviving because of these kind of drugs. But there's a problem. These drugs are very expensive. So if I said to you, on average, to treat one patient for 1 year is in the order of about £75,000 per patient. And we're treating thousands of patients in the UK and worldwide. And the issue for us is that actually not all those patients are benefiting. Melanoma is one of the most immunogenic, immune-responsive cancers, but we're using these drugs in lots of other areas where the response rates are much, much lower. And if you look at this curve, you can see, if you go across from the bottom, 2011, 2022, these are the indications, the availability now, the approvals for using immunotherapy across multiple cancer types over time. But if you look at the response rate, so these are the different types of cancers on the left-hand side, and on the top, the numbers are the proportion of patients responding to treatment. Ideally, you want 100% of people responding to the treatment we're giving. Unfortunately, even with melanoma, we're only reaching about 50%, as I said. So that means that we're using these drugs with lots of people, but only a small proportion of them are going to benefit. So that's a major issue in terms of resources, literally pounds, shillings, and pence. But also there's a burden on patients because these drugs have complex side effects some of which are life-threatening and definitely life-changing. And if I give you an example of one of our patients who was treated back in 2017 now, this is a timeline of her time on when she started treatment. The little red flags on the top, she had 2 cycles of Ipi-Nivo. And after her second cycle of Ipi-Nivo, everything went pear-shaped. So she developed thyroid dysfunction, a really nasty rash, and then she had awful diarrhea and colitis. And she came into hospital and she spent nearly 3 months in hospital being managed for really awful colitis. She didn't ultimately need surgery, which was great. Some people do. During that time, her liver function went off. We managed that, and ultimately we got her home. But shortly after she went home, she started developing a really nasty erosive arthropathy. So she is now alive. She is cured of her cancer, but she is living with chronic arthritis, and she's actually moved to Spain because the weather's better. So that's the kind of price that people pay for the benefits of these kind of drugs. And we're still in the early days of learning about these drugs. So what we're now wanting to do is, we've got these drugs, can we personalize care? And this is when we use what we call biomarkers. And we can use biomarkers to try and identify people who might be at risk of cancer, who might have a particular form of cancer, who, if they've had cancer, is it going to come back? These kind of things. And the one we're particularly interested in the context of immunotherapy is, can we predict those people who are more likely to benefit or more likely to have toxicity, so that we can actually tailor the right treatment to the right people and avoid giving drugs to people who we think it's not going to work in. What do we use for biomarkers? Well, we use any kind of material we can get our hands on. And the classic things will be tumor tissue. So if we can get a piece of the cancer from that patient, can we understand the molecular and the characteristics of that? And we send it to the pathologists and the genetics guys, and they do lots of tests on that nowadays. We can take blood, which is readily available. We can look in blood for all sorts of things. And nowadays we're looking at other things like stool and urine and anything, literally, any product of the body or the tumour that we can start to study, we will study. But lots of people have been working in this arena, and at the moment, we're not really making a huge amount of progress in terms of those biomarkers. A lot of resources are going in, and a lot of people are working independently. So here you can see we're trying to identify, if we've got a whole bunch of people in my clinic, who are going to be the responders, those who are going to be unresponsive, and those are going to run into toxicity. And we can access these materials, but how are we going to find those biomarkers? And the challenges we have is that biomarkers research is not straightforward. If you think about a piece of tumor tissue, it's not all homogeneous. If you look at it under the microscope, you see different types of cells. And so if you take a biopsy, do we know if that's representative of the whole tumor? And if we take a blood sample, in a blood sample there are cells, there's protein, there's DNA. Which bit of that blood is important? And what timing of taking that sample is important. If we take a tumour sample at somebody's diagnosis of melanoma, the time they start treating for metastatic disease may be hugely different. And is that original piece of tissue going to be informative at the time of starting treatment, or do we need another biopsy? There are lots of these kind of questions that are proving very challenging. So the vision of Manifest is that we want to harmonise all this. We want to create a single national platform where we bring together all the experts working in this field so that we can actually use all our clever technologies in one platform and generate, hopefully, a comprehensive analysis of the various samples that we can get our hands on, and hopefully inform clinicians better as to how to manage those patients. So ultimately, could we envisage at the end of this exercise creating a biomarker report that could be handed over to the clinician and the patient to say, actually, this is the person who is going to benefit, this person isn't, we should be thinking about offering them something different, or this person is likely to have problems with toxicity, we should be avoiding X, Y, Z. So this is where we want to go. That's the vision. So Manifest is a consortium that was funded last year, and it's based upon some funding which was made available by the Medical Research Council that has something called the Office for Life Sciences. And they offered up some funding, and SAMRA coordinated a collaborative. So this is a map of the people or the centres that are engaged. This is clinical teams and academic units who are coming together across the country. And there is a huge amount of industry engagement. So you can see here a lot of additional matched funding that industry is coming into this because they have amazing capabilities when it comes to technologies and basic science. So this is a real consortium, a partnership of multiple stakeholders that hopefully will pay off if we work together rather than all this kind of historical parallel working. So I'm not going to go into this in detail, but basically just to demonstrate that there are several work streams which are focusing around specific technologies that we know are relevant in the biomarker area. And what we hope is that we will be able to comprehensively bring that information together in terms of understanding and interpreting the biomarker data. We may be able to create predictive models. We may be able to create that comprehensive report that could be clinically useful. We hope that because this technology and this platform is going to be running over a number of years, that we can incorporate new technologies as they come on board. We may be able to understand more about the basic science of immunotherapy resistance, and we may even be able to identify new targets for new therapies in the future. So lots of hope and optimism as we get going. So how's this proceeding? As I said, we started last year. The kickoff was August last year. And there's a scientific advisory board, data management group. And what we've done is we've brought together people who, like myself, already have research studies in this area. We already have patients who've consented to allowing us to look at their data, collect samples relevant to biomarkers, and these patients are all receiving standard of care immunotherapy. So we're bringing people like myself together where we can actually share our information and our data and our samples. So we'll do some retrospective work, and that will include about 3,000 patients. And then going forward, we're planning to recruit a further 3,000 patients prospectively, again, through these projects that are already up and running, but also a Manifest-specific protocol. And then when we've done all the exploratory work, we will validate and test our hypotheses and our findings in what we call a validation cohort. So this will be a sort of a 5-year project at least. And the cohorts that are particularly of relevance are here, we've listed down here. Melanoma is one of them because melanoma is one of the cancer types where immunotherapy is being used most, but also has a role in these other cancers, renal cancer, bladder cancer, and breast cancer in particular. And there are a variety, again, of those kind of technologies and the work streams that will be focusing on different biomarker projects. And then prospectively, you can see here, this is just an example of all the kind of samples that we're going to be collecting. So this is tissue, blood, different parts of blood, cells, liquid, DNA, and then also things like stool, and we can talk a little bit about that in a minute. And the variety, again, of the different work packages, all headed up by some of the leading experts in the country and beyond, actually. So some of these people are international world leaders in their field. So it's a really, really exciting project. And down here you can see the key NHS trusts, which are at the moment engaged, but hopefully by the time we get to the prospective element, we will be bringing on board more trusts if we can encourage them to be involved. And at the end of the project, we are hoping that there will be a legacy and there will be sustainability. So if this goes really well, it will basically be self-sustaining. We should be able to, because we're bringing in these links between academia and industry and the clinical networks to try and continue to work and maybe change the way that we manage patients at the moment when they receive immunotherapy. So if we are generating these kind of biomarker reports, do we need an immunotherapy tumor board that can inform and work with the clinicians and the patients to make sure that we really do focus in on personalising the treatment of the future. So I mentioned that this involves melanoma, and the title of this was Collaboration is Key. So I just want to demonstrate how we got involved from the perspective of melanoma and MITRE, which is the study that I lead, which is very much a collaborative as in the Manifest consortium. So I think you've heard before in this forum about the importance of the microbiome or the increasing interest in it. We believe that it is a biomarker predicting for immunotherapy response and toxicity. And for those of you who haven't heard about this so much, our body is an ecosystem. We are literally integrated with a variety of organisms, mainly bacteria, but various other things, fungi, viruses, and they live in various parts of our body, in our gut, on our skin, in our lungs, and they are really important in maintaining our body health. And when the microbiome is disrupted, health can be disrupted, so it can be involved in poor health and disease. And you can see here, these are just some of the conditions that are linked with the microbiome, many of them associated with inflammation and immunity, and they include cancer. And interestingly, there is growing evidence to suggest that the microbiome is linked to the response to immunotherapy. And just to demonstrate on the right-hand side of the slide that you can see, this was some really lovely data that sort of kicked off a lot of interest a number of years ago, where a group of scientists grew in a mouse model melanoma and treated them with an anti-PD-1, a checkpoint inhibitor. And what they did was that they introduced into the gut of those mice some stool, either taken from a melanoma patient who had responded to immunotherapy or one that hadn't. And if they introduced the stool from the patient who had responded, the mouse responded. But if they put in the stool from the non-responding patient, the mouse did not respond. The tumor just grew rapidly. And now this kind of animal experiment is now moving into the clinic, and there's some early phase data to suggest that these kind of things called fecal microbial transfer can actually be used to modify and manipulate response to immunotherapy in people. So it's a really exciting area. From my perspective, I live in Cambridge, I live and work in Cambridge, and down the road from us is the Wellcome Sanger Institute. Trevor Lawley is a scientist there, he's a group leader, and he's a world leader in microbiome research. And his technology is a much more detailed analysis of stool. I hope you're not getting all squeamish first thing in the morning talking about stool, but it's wonderful stuff. And he basically collects all these stool samples, cultures out the bacteria, and then does very deep sequencing at the molecular level to understand exactly what strains of bacteria exist there. And that's much more detailed analysis than many of the people do in this field. And we got together in 2017 and started collecting stool. We were a little bit anxious about whether patients would engage in this, but they've been fabulous. We were collecting it from patients receiving checkpoint inhibitors routinely. We were able to look at the bacterial signatures of those patients who were either responding or not responding to their immunotherapy. And this looks a busy slide, but I'm just going to point to the middle piece of this graph. So from our first 50 patients, we were able to pick out a group of about 9 bacteria, and these are the bacteria circled in the green circle that were consistently linked to those patients who were responding to immunotherapy. And what was interesting, although many people had already published this kind of work, they were very much focusing on 1 or 2 specific bacteria, and they were all coming up with very different conclusions. What we did was we went and reanalyzed all the data that was in the public domain, and were able to, on the left-hand side, you can see separate out non-responders and responders across multiple international data sets. So we thought that was really exciting, and we've now put these bacteria into a capsule. On the right-hand side, you can see this is, again, in a mouse model, growing that melanoma in a mouse and treating either with PD-1, a checkpoint inhibitor, or checkpoint inhibitor plus this product, MBO97. You can see the bottom, the purple line, are the mice who were treated with both the anti-PD-1 and the MBO97. You can see that in that situation, the tumour doesn't grow very much. So we're very excited about that, and that is now in a human clinical trial, which is currently recruiting. So back to the biomarker work. Although we've done this initial work, we've got to validate it and really make sure that it works and is relevant across multiple types of cancer, which links us back into Manifest. So we're continuing to recruit people with melanoma, renal, lung, and now bladder cancer, collecting their stool samples and doing detailed microbiome analysis, but we're also collecting other samples. So we're collecting blood and we're collecting tissue, but we don't have ourselves within our team the expertise to analyze those samples. And so Manifest has come along at such a good time for us because we can now coordinate, collaborate with them and utilize this platform so that rather than us trying to think, well, how are we going to do these kind of complex experiments? We don't have the expertise, we don't have the funding. We can join with Manifest and basically say, hey, here are all our samples, here's all our data, you've got all this wonderful expertise, now you can do the analysis for us and we will work with you. And then we can link up the work that they do with the microbiome work that we're doing. And in fact, Trevor Lawley is very much leading the microbiome element of the Manifest program. So this has been a really good opportunity, really good evidence of how we can bring scientists, clinicians together to work together, hopefully to better effect. So the ultimate aim that we have, the shared goals of both teams, is that we really do want to personalize the future care of our patients by predicting response, predicting toxicity, avoiding unnecessary exposure to potentially ineffective, toxic and expensive drugs. And then we want to be moving into the intervention area where we can utilize these outputs in order to actually modify and improve outcomes for our patients. And at the moment, for example, turning a non-responder into a responder by adding in, for example, our microbiome product. And so that's the conclusion. I hope I've illustrated or tried to explain this complex title to you. What is Manifest? So if you hear that name, you will understand what it's all about. And we hope that we will be recruiting patients in the near future, specifically to the Manifest protocol. It's very much a collaboration, and it's also very much a collaboration with you, the patients and the public. And there is a patient group involved as well in order to make sure that they keep us online and we focus on the things that are important to you. So I'm going to stop there. Thank you.